Hi, I am Dhrubajyoti Maji, a senior research fellow in CBMS department under Professor Ranjit Biswas. Today I am going to talk about my project Structure and Dynamics of Glucose Ethylene Glycol Cryoprotectant System, a molecular dynamic study. So what is a cryoprotectant? A cryoprotectant is a substance which is used to protect biological moieties from cold denaturation. How does a cryoprotectant work? There are four supposed pathways, like it can depress the ice nucleation, can promote glass transition, preserve the cell membrane, or stabilize the native structure of protein. Depending upon their active sites, cryoprotectants can be classified into two categories: permeable and non-permeable. Permeable cryoprotectants can diffuse through cell membrane and can act as both intra and extracellular space. Alcohols, sulfoxides, amides are common examples of permeable cryoprotectants. On the other hand, sugars, sugar alcohols, polymers are non-permeable cryoprotectants. Here we have taken one permeable cryoprotectant, ethylene glycol, and a non-permeable cryoprotectant, glucose, and we mix together them, and we want to see what are the interactions occurring between them, that is how they behave in presence of each other. So we have taken a pure ethylene glycol solution of 3 ethylene glycol glucose mixtures containing. So we have taken a pure ethylene glycol and 3 ethylene glycol glucose mixtures containing glucose weight percentage of 20, 30 and 40. Here we present the simulated and experimental densities, and a fair agreement between these values ensures us about the validity of our simulation protocol. For structural study, we have calculated radial distribution function between different atom pairs of ethylene glycol molecules. This is the RDA for oxygen-oxygen atom pairs, and this sharp peak appears due to the hydrogen bonding interaction between Gauss conformers of ethylene glycol molecules, and this is due to the intermolecular interaction of between oxygen atom sites where no hydrogen bonding is involved. This is for oxygen and hydroxyl hydrogen interactions, and this sharp peak is the characteristic hydrogen bond peak, and decrease in first peak height and slight shifting of peak maxima depicts the inhibition of glucose molecules into the hydrogen bonding network of ethylene glycol molecules. Here we present the RDF of carbon-carbon and carbon-oxygen atom pairs. And this is the static structure factor of ethylene glycol in pure form and in solutions also. We can see that the principal peak appears at 0.4 nanometer, and which is almost fall in the region of same length scale of the peak maxima of carbon-carbon and carbon-oxygen RDFs. That is, the peak decrease in the peak height means the decrease in the correlations of carbon-carbon and carbon-oxygen interaction. That strengthens the fact that invasion of glucose molecules into the ethylene glycol network, which we have seen earlier. Here is the radial distribution function between oxygen and hydroxyl oxygen hydrogen atom pairs between glucose-glucose and glucose-ethylene glycol molecules. Here we see no significant change, barring slight decrease in the main peak height with increase in glucose concentration, and this main peak is the characteristic hydrogen bond peak. Both ethylene glycol and glucose have high propensity to form hydrogen bonds. Ethylene glycol has 2 donor and 2 acceptor sites, whereas for glucose there are 5 donor and 6 acceptor sites. So for a ethylene glycol glucose mixtures, there are essentially 2 types of interaction. One is interspecies, that is the interaction between ethylene glycol and glucose, and another is intraspecies, that is the interaction between ethylene glycol and glucose with themselves. And both ethylene glycol and glucose are capable of forming intermolecular as well as intramolecular hydrogen bonding. To know about the hydrogen bond network, we have calculated number of hydrogen bonds per molecule. For ethylene glycol, we have seen that the intramolecular hydrogen bonds per ethylene glycol molecules remain almost unchanged across solution. But the intermolecular hydrogen bond and also the overall, that is the total number of hydrogen bonds per ethylene glycol molecule with ethylene glycol, decreases with increase in glucose concentration in the solution. Whereas for glucose molecules, here also the intermolecular hydrogen bonds remains almost unchanged. But the number of intermolecular hydrogen bonds and hence number of total hydrogen bonds between glucose molecules increases with concentration. So what happens in the interspecies hydrogen bonding network? We can see that the number of hydrogen bonds per ethylene glycol molecules with glucose increases with increasing glucose content in the solution. But the reverse trend is observed in case of glucose molecules. So what is the overall scenario? The overall scenario is the total number of hydrogen bonds per ethylene glycol molecule remains almost unchanged. But there is slight increase in total number of hydrogen bonds for glucose molecules with increasing glucose content in the mixture. This can be again observed from these figures. Here we can see that the glucose-glucose interaction increases and glucose-ethylene glycol interaction decreases with increasing the concentration, and this rate is much more than this one, and the overall number of hydrogen bonds increases. For ethylene glycol molecules, the ethylene glycol and glucose interaction increases and ethylene glycol interaction slightly decreases, and these two factors almost nullify each other and the total number of hydrogen bonds remains almost unchanged. So from this study we can say that both ethylene glycol and glucose molecules prefer glucose molecules at their hydrogen bonding partner. For dynamical aspects, we have calculated the collective dipole moment autocorrelation functions. The timescales obtained here are comparable with the timescales obtained in dielectric measurements. Here we can see that the long timescale goes from 200 picosecond to almost 2.5 nanosecond. So this enhancement of this time scale may be due to the glucose-ethylene interaction and glucose-glucose interactions. A 40 picosecond time scale is observed in all cases. Hydrogen bonding interaction and reorientation of molecules can significantly contribute to these time scales. So we have calculated the reorientational correlation function of rank 1, and we have seen here also the same trend, that is the long time scale getting slower and slower with increasing glucose content, and also 40 to 50 picosecond time scale appears which is comparable with the 40 picosecond component of the dipole autocorrelation function decay. For structural hydrogen bonding relaxation function, we have also seen the waiting time increases with increasing glucose concentration, that is also the rotation time increases, and this intermediate time scale is well accordance with the earlier observed sub 50 picosecond time scale, and also a 10 picosecond time scale appears for each case, and this is a further investigation needed for the origin of this time scale. So as a conclusion, what we have done so far, we want to study the interaction, both structural and dynamical, between ethylene glycol and a mixture of ethylene glycol and glucose molecules. Our structural study reveals the propensity of forming hydrogen bonds for both glucose and ethylene glycol is very high, and both ethylene glycol and glucose prefers glucose as their hydrogen bonding partner. That is, the ethylene glycol network gets ruptured by incoming of glucose molecules into the solution. From dynamical study, we have seen that the dynamics is getting slower and slower with increasing glucose content in the system. And as a future study, we want to work on the preservation of biomolecules. That is, we want to see the interaction of biomolecules in presence of these cryoprotectants in sub-zero temperature. I want to acknowledge my supervisor, Professor Ranjit Biswas, for his constant guidance, and I also thank my center, SNBN CBS, DST Inspire, Jadavpur University, and my lab members. Thank you all for your kind presence and attention.